Hello, everybody. So my name is Martin Hanichinec. I'm a PhD student of Professor Tennyson's. I'm working on a project of optimizing chemistry sets for plasma modeling and closely collaborating with Quantemol on it. So I'll try my best to present a gist of my work in this short talk. So the title of the talk is Optimizing Chemistry Set, but the talk is more generally about reduction of chemistry set size, and more specifically reduction in number of species. So Dr. Moore showed you how you can go about like designing chemistry sets. But yeah, I'm gonna be talking about reduction their sizes when you find a chemistry set which is like too big for your application. So I thought I would start with a little motivation for the presented work. So as you know, and as Sebastian told you already, like no matter what sort of dimensionality of your numerical model of plasma, or no matter what sort of underlying physics in the model you're capturing, pretty much every model is sort of centered about the chemistry set, or it might be called a kinetic scheme, reaction set, kinetic mechanism. It's not very well united, the nomenclature. But yeah, anyway, these sort of chemistry sets, they are nothing more than set of reactions between species tracked explicitly by the model, and kinetic data assigned to every reaction. So you can find these chemistry sets, you can find them published online. You can find them for various gas mixtures, but they do not come without problems. And one of the problems is so often the size of the set. So some chemistry sets might contain up to 100 species, many thousand reactions, and this can cause great problems with computational cost if your model of choice is let's say higher dimensional, or captures like more refined and subtle physics. But in fact, almost all published chemistry sets contain redundant species and redundant reactions for any sort of narrow application. And yeah, this work is about, or this presentation is about reducing chemistry sets by identifying these redundant species and eliminating them out of the set. So if we start with a chemistry set which is sufficiently detailed for our modeling application, we can ask a question about which species and reactions are in fact redundant. But this question, it does not have an absolute answer, because in fact, when we are talking about redundant species, we need to relate the redundancy of them to the specific set of conditions and to a specific set of model outputs we are actually trying to replicate, we're actually interested in. So for that, in this work, we're defining notions of species of interest and reduction conditions. So basically, this means that the reduced model we're trying to, or the reduced chemistry set we're trying to find or build, it needs to replicate the densities of all the species of interest under the reduction conditions. So that's all that's to it. And yeah, important thing is both species of interest and reduction conditions, they are ultimately user defined. So let me try to start with an example to just demonstrate and drive it home a bit more. I'm going to show you a couple of slides about reduction I did on N2H2 chemistry set. So I started with a detailed reaction set published by Hong. It contains 42 species in total and 408 reactions. So the species are listed here in the table, but it's not really that important, so I'm gonna carry on. In this reduction, we define species of interest as NH, NH2, and NH3 radicals, and the reduction conditions were defined as listed here as well. So this was low pressure, 9 pascals, it was room temperature, and certain feed gas ratio and certain power density as well. So all of these are actually relevant for an application of low pressure plasma ammonia synthesis. So we know from literature that ammonia is mostly produced by NHX radicals, and yeah, these plasma conditions were also taken from literature as sort of demonstrative conditions for low pressure and ammonia synthesis. So for these conditions and species of interest, we actually identified that 23 species out of those 42 were redundant for these conditions. And this resulted in a significant reduction in number of species and reactions in the chemistry set. So we dropped number of species from 42 to 19 and number of reactions from 408 to 91. So you can see this is a significant reduction. And on the right hand side, you can also see that on the comparison of modeling results with detailed and reduced chemistry set, the densities of all the species of interest we defined before were indeed almost perfectly replicated with the reduced set. So of course, this is nice, but the real key here is to be able to do this thing dynamically and for any conditions or species of interest. And yeah, in the rest of these slides, I'll try to outline the method we're using for this. So the method revolves around creating a species ranking. So we can rank the species according to their importance for modeling all the species of interest. I'll try to go through this quickly. We start with detailed chemistry set. In our case, this was N2H2 in our simple example. And we have reduction conditions which we define. And yeah, again, for our, okay, so these are slightly different than before, but yeah, this is just an example. So we start with these two and we feed those two to a global model of plasma. So in my work, I use a plasma global model, which I developed so specifically for this. So it's a Quantemol in-house model. It's the same model the QDB website uses and the QDB application will use. So it's the same model as Sebastian showed you in previous presentation. So the global model in turn gives us densities and reaction rates of all the species and reactions. And we can use this data or these results to build a chemistry graph. So this is just a graph with species represented by nodes, and directed edges from node to node are representing asymmetric direct relations between species. So the edges are weighted by direct interaction coefficients. And the weight of any edge between two species is a function basically of reaction rates of all the species involving both of these species, of all the reactions involving both of these species. So basically, two species only have an edge if they from one another if they share at least one reaction together. But of course, species influence each other also if they do not share any reactions together. So therefore, we need to define indirect coupling coefficients. And these are defined for each two species, and they are functions of paths or all the paths leading from one to another. Let me divert a little bit just to explain this in a little bit detail. There's not enough time to go through all the details, but very briefly, the direct interaction coefficients are basically just adopted from DRG theory. That's direct relation graph theory. That's well-established theory which can be found in literature. I'm showing a citation here. And the indirect coupling coefficients, I based them on Dijkstra's shortest path graph search. So as I said, not enough time to go through the detail, but yeah, that was just a brief diversion. So if we go back to the species ranking flow, we can now introduce the species of interest to the picture. So in our example, it was NH, NH2, and NH3. And then we can fairly straightforwardly define species ranking according to so for each species, ranking score will be uh, basically dependent on coupling coefficients from that species to all of the species of interest. So that's just a very brief gist of the species ranking. And yeah, by the way, for anybody interested in more details, there'll be a paper in PSST describing this method, which was actually literally yesterday accepted, and it should be online now, as I understand. So, okay, finally, we have ranked the species, and it can be said that the highest ranked species are likely to be essential species for our application, while the lower ranked species, or these bottom dwelling species, are in turn likely to be redundant for our applications. So how can we do the chemistry set reduction now with this species ranking? So what I'm using here is what I call species-oriented ranking-based iterative reduction method. And the method is sort of described by this flow chart. So what we do is we start with the we start with okay, let me turn on the pointer. Okay, so we start with detailed chemistry. We start with the reduction conditions and with plasma model. Again, in this application, it was our in-house global model. And we basically solve the plasma model with this detailed chemistry set. And then we build the species ranking, as I showed before. And once we have this species ranking, we enter this iterative reduction loop, where in each iteration, we basically eliminate the lowest ranked species, rerun the global model, evaluate the error, the reduction induced, and if the error on the species of interest is acceptable to us, then we go on to the second iteration. If it's not, then we need to reinstall the species back into chemistry. And we just go on and go on until we meet some sort of termination condition, which can be defined in several ways. And yeah, that's it. Once the loop finishes, you're basically guaranteed that the reduced model replicates the species of interest within an acceptable error margin. So that's a method we used for chemistry reduction, or for species-oriented skeletal reduction of chemistry sets. And okay, as a final slide, just to expand on this a little bit, just want to mention that often the reduced chemistry set needs to be valid over a range of conditions rather than a single set of reduction conditions only. So the method here is to sample multiple sets of reduction conditions from their parameter space. So if we want ranges of pressure, ranges of power, ranges of, I don't know, feed gases or gas temperature, we basically can do some sort of design of experiment stuff to get several sets of reduction conditions from this parameter space. And then we can simply just run the reduction algorithm on each set separately. And then only the species which are identified as redundant for every single set of sample reduction conditions, only those will be removed to form the reduced set. So that's pretty much all I have prepared here. And I see that I am running out of time anyway. So that is it. I'll thank you for your attention. If there are any questions, I'll be happy to answer. And yeah, again, let me mention that if you're interested in details, check out the paper, which has been accepted to PSST. And yeah, I put a link to that paper here. So that should be online now. Okay, thank you.